So as Reza already mentioned, I'm Yaser, and I'm group leader at Patrick Johnston Centre for Cancer Research. So my research group is mainly working on chromatin biology, cell biology, epigenetics, and stem cells, and cancer, a lot of things. But I think recently the entire field, including, of course, my group, has become interested in the application of AI, as you can imagine, like everyone else. And this is really taking over so many fields, including very basic molecular biology that we are doing. So I thought I was talking with Reza, over coffee, and we thought maybe this is something would be interesting for you also to learn a little bit about this field. And I'm super actually excited to see how the AI application is developing in our field, and I think it's just the right time now. So what we have been doing, well, not personally me, but the field has been working for 50, 60 years on this topic. And I think now it's the time where, computer, machine learning, AI has really come to help us to address that key question that we have been trying to address for so long time. So please interrupt me during the presentation. It's a probably different field, of course, from what you guys are doing. So I would like to have more sort of interactive talk where if you have any question or if something is not clear, just interrupt me during the lecture and we can address those questions. So you heard about, everywhere now in the news, right, that AI is super important in different fields, including biology, chemistry, and so on. And probably you had some lectures already, outlining some of these applications in medicine and health. Some applications that are super exciting now is, for example, to predict the molecular structures of protein, macromolecules, and then to use, for example, those predicted structures for in silico drug discovery, right? People had to screen thousands of compounds in the lab to find few compounds that work better. But now with, this AI in silico learning algorithms, you can do many of these filtering in your computer and then test some limited set of those experimentally. Obviously, you heard in news that people are now using AI to look at, for instance, CT scan from cancer patients, trying to identify tumors which are more difficult, and actually it has even more precision and accuracy than pathologists and human eye. And this has also been applied, for example, in screening tumor sections, looking at cancer cells and trying to identify the abnormal cells in different samples, including, for example, samples taken from blood, from urine, and so on. So many of these applications can be done, of course, by human eye, but AI has been trained to have more accuracy and more precision. So what I'm going to talk today is slightly different, and that's also sort of pattern recognition application, but not really looking at cells and cell morphology and try to understand the abnormal cells. But here we are looking at DNA sequence. So my talk will be to understand how we can screen DNA sequences to identify regions in the genome, which we think they are important for gene regulation and gene expression. And so we call those regulatory elements or enhancers. And the field has been trying to identify these sequences for 50, 60 years using many different algorithms. But now I think with machine learning and AI, we are getting there. So this is the focus of the talk, how we can identify regions in the genome based on DNA sequence and other features in which we think they have some functions, in this case, regulatory functions. And so the learning outcomes that I would like to have at the end is basically describe the logic of regulatory elements. So this is a high-level overview of what these regulatory elements are. You're not biologists, so I'm not expecting you to know all the molecular details, but this is more a high-level overview of the basics of enhancer and gene regulatory elements. And then describe the current challenges in identifying and understanding these elements. Try to explain how we can use deep learning models to annotate enhancers. And finally, how we can use now all this knowledge to actually synthesize enhancer de novo from scratch. So these are the 4 main objectives. Again, I know that your expertise is completely different from what I will talk today. So I will start from the very basics, and that is, as you know, well, human body is composed of different organs, and each organ has tissues, and in each tissue we have multiple cell types. And within each cell, you have the DNA, which is the coding sequence. That's the program, right? In which it is actually located in a small, tiny structure, which is called nucleus. So if you think about cell as a computer, nucleus is the CPU, right? And the DNA is the program, is the code. So all the information that we need is within this. And it's super nice. So yesterday we had this solar eclipse, right? If you stretch the DNA from one single cell, it will be 2 meters. And so this 2-meter molecule is actually compacted in a very, very tiny space, which is in 5 micrometer in diameter, and that's the nucleus. Now, if you take the DNA from all our cells, which is about 30 trillion cells, and you just stretch them, right? The distance will be almost 400 times the distance between Earth and Sun. So you can imagine what molecule we have in our body, and that's one human. So now you multiply that by 7 billion or even more. So that's amazing, right? How nature could compact this 2-meter DNA in such a small space. And this is done by folding DNA in different levels, so you can compact it in a very, very tiny space. So this is the real life image of a cell and the real life image of a nucleus, right? So this is the nucleus in which the DNA is compacted. And now in normal dividing cells, you don't see the DNA is here basically, and this is the sort of gene expression factories, right, where you have high expression of genes. That's why it's more darker. So in normal dividing cells, you don't see the DNA. It's sort of it's a string. It's like a spaghetti, right? But during cell division, then it becomes compact into these structures called the chromosomes. And then within this chromosome, of course, you have the DNA, and this is the real life image of a DNA. So the way that I usually describe this is you have huge information compacted in different chromosomes. So you can consider all these 23 pair of chromosomes as a library of books. And then each book you can consider it as a chromosome. And then the text within each book is the analogy of the DNA sequence. Now the question is, which has been one of the biggest actually questions in biology, and that is, all cells in our body have exactly the same DNA sequence. So how come that one DNA sequence can generate so many different cell types, skin cells, neural cells, blood cells? If the DNA, if the program is exactly the same, you should expect one outcome. But we have thousands of different cell types with different functions. So how is this regulated? And this is the central question in biology. Now, one way to explain this is to understand how the DNA is actually organized. And that is, if you think about DNA as a coding sequence, you have regions which are called genes. And those genes will make proteins, for example, globin, right, that is present in your red blood cells, or keratin protein, right, that's present on skin cells. A protein called myosin, which is present, for example, in muscle cells, and so on. So these are genes that encode proteins. But why, for example, the globin gene is expressed in red blood cells and not in brain cells, right? So there you have these regions which are not coding proteins that are called regulatory elements. And their function is basically to control the expression of the target gene. So these are called enhancers or regulatory elements. And they are super nice because they can actually control it's like a volume control. So they can control the strength and the specificity of target gene expression. And it's funny to know that if you take our genome, it's actually only very tiny percentage of our genome encode proteins. So only tiny percent are genes, which is 2%. And 98% of your DNA is actually, they are not genes, they are all these regulatory elements, structural elements, and so on. And if you compare, for example, tomato with human, we almost share similar number of genes. Nevertheless, the complexity of human cells is much, much higher than complexity of tomato, of course. And that's because if you look at, for example, the number of regulatory elements in human cells, there are almost 10 times more than the tomato cells. So the complexity is not always determined by how many genes we have, but by how many and how these regulatory elements are controlling our gene expression. Now, the funny thing is that, so as I mentioned, enhancers and these regulatory elements have two different functions. One is to control the level of gene expression. So again, it's like a volume control. You can have 10% expression, so 10% proteins or 100%. But nature also found another way to even control this more. So one enhancer can function in different levels. But nature also found that if you combine now different enhancers or different regulatory elements, then you can also have another sort of level of control, right? Because now each enhancer can be controlled separately, and the combination of, let's say, different enhancers can give you different outcome. And on average, it's estimated to have, for one gene to have around 4, 5 enhancers. And for some very important genes, we also have up to 30, 40 different enhancers. So that gives you, an extra layer of complexity to control gene expression. The other function, the second very important function of enhancers, is that they also control the specificity of gene expression. So as I mentioned, your globin gene is not expressed in brain cells. And like, keratin is expressed usually in hair and skin, is not expressed in, I don't know, muscle cells. And the way that this is done is that these enhancers are cell type and cell state specific. So, for example, if you take red blood cells, enhancers that regulate the globin gene are active. But if you take the neural cells, those enhancers are inactive. And so that's why this gene is not expressed. And in the neural cells, for example, you have this other gene, which is neural specific, in which those enhancers then active. So that's how you control where and when genes are expressed. For simplicity, I just put here one product per gene, right? That's one. And for simplicity, I am also basically presenting the enhancers as active or inactive. But you can also have different levels of activity and different activity in different enhancers. But for simplicity, let's say all the enhancers are not active in neural cells and all enhancers are active in red blood cell, leading to either switch on or off of the target gene, in this case, the globin. But this is the extreme example. The way that actually in reality it functions is that you have, again, combination of different activity of different enhancers, which leads to different levels of expression in different cell types. Obviously, you also have genes which, as I mentioned, for example, globin is not expressing in our brain cells. So that's completely on and off. So the entire set of enhancers are either active or inactive in this case. Yeah. So now let's dive into just try to understand what enhancers are, right? So how these enhancers are active. When I say active and inactive, how is that regulated? So think about enhancers as docking sites for some proteins, which are called transcription factors. So these are the regulators that bind enhancers and activate them, right? So the term transcription factor is coming from the term transcription. So the outcome of gene expression is transcription of the target gene. And so that's why they are called transcription factors. So enhancers are basically just the docking sites of these transcription factors. Again, for simplicity, I just put one transcription factor per enhancer, but actually in reality, you have multiple transcription factors that bind one enhancer. And the combination of these transcription factors is important. So now you see that this is actually the way why the enhancer can have different spectrum of activity. Because now you can have binding of one transcription factor, 2, 3, or 4 to one enhancer. And so that combination will determine the activity of the enhancer. So that's how mechanistically these enhancers are inactive or active or in between. And the second complexity, as I mentioned now, is that you also have different enhancers. There are multiple levels of how you can have a spectrum of activity for target genes. Multiple enhancers, multiple transcription factor per enhancer. And the other thing to remember is that, so when I say transcription factor, you have different transcription factors binding to one enhancer, and that's because each transcription factor has its own target site. So each transcription factor has a specific DNA sequence that can recognize and can bind. And these are called motifs. So within each enhancer, you have multiple motifs in which are basically the recognition sites for these transcription factors. So it has been, this field has been working on this subject for so long to understand which sequences are recognized by which transcription factor. And to some extent, we know for many transcription factors what are the target sites, but for many transcription factors, we still don't. And one reason is that, so I put like one example there that you see, for example, the probability of having, different nucleotides at different regions is shown in the height of that graph. So that means, for example, at position 2, 3, sorry, in this case, you have more probability of having T versus others, and so on. But it's not one-to-one correlation. So for some transcription factor, we can exactly say this is the sequence that will be recognized by this transcription factor. But for others, it's not a clear cut. And the reason is that transcription factors can also cooperate. So they can help each other to bind the target sequence. So that means that depends on the other transcription factors present in proximity and the other motifs present next to your motif. That can influence how strong your transcription factor will bind to this particular sequence. So it's the sequence, sure, but also the context. So the surrounding sequences, that's second level, and the presence or absence of the regulatory transcription factors. So you can have the motifs nearby, but if the transcription factor is not there, well, it will be not active. So it depends on the sequence complexity and depends on the complexity of the transcription factors that are present in the cell. You can have less or more binding of a particular transcription factor. So this is the logic that people have been trying to understand for a long time. And there are now databases, right, where you can actually find like the motifs for all transcription factors that we know. And you see things like this, which says, well, this is the likely sequence. So by now, I think you started to realize that actually the enhancer code or the DNA code, it's a sort of language, what do you call it? It's like a language, that's like a grammar, right? So if you think about gene expression as a text or as a poem, let's say, right? You can divide that text into paragraphs, which in this case, it will be multiple enhancers functioning together. And then you can divide, for example, the paragraph into different sentences. So in this case, each sentence will be one enhancer. And then each sentence into different words. And in this case, each word corresponds to one motif. And then each motif into nucleotides or sequence, which is the letters, basically. So it's a language model anyway, right? And that's why it's super nice now to use all this, machine learning and AI to, similar to what ChatGPT is doing, try to understand this language. So that's how I'm going to continue from now. All clear so far? Is this analogy clear? Yeah. So now let's explain a little bit on how we measure enhancer activity, because that's something we will need later on. So when I say, enhancer is more active, it means that it can enhance the target gene expression more compared to other enhancers, right? And we can measure this experimentally. So that's how we measure the enhancer activity. And that is either by measuring the transcription of target gene, and we have experimental ways to measure the RNA, the transcription, or we can do this. This is done for one gene, for one enhancer, one gene. But if you have thousands of enhancers, and by the way, in our genome we have around 100,000 enhancers. So if you want to measure the activity of 100,000 enhancers, then it will be more difficult. So then we have different techniques, like high throughput techniques. I just put one example there, where you can actually measure the activity of thousands of enhancers in one experiment. And now you have a readout, right? So now you have all your enhancer sequences from 1 to 100,000, and you have an output saying that this enhancer has this level of activity in this particular cell type. We also have another way of measuring enhancer activity, and that is the features of enhancers. So what people found is that those sequences, so again, for simplicity, I showed DNA as like a naked DNA, right? But actually, in reality, DNA is not just a DNA. It's a DNA that is wrapped around proteins, right? Histones, other transcription factors. So it's like a party. There are a lot of things going on. So when you see DNA, well, it's not really DNA only. It's a complex of different proteins. Now, the funny thing is that at enhancers, people found that there are specific features. So there are chemical modifications, either the proteins or DNA or transcription factors at enhancer sites. And so those features are super important for us because actually we can say, oh, okay, so I found this chemical modification here, so it must mean that, there is an enhancer located here, and then we can say it's active or inactive. Now, this is an important point. These chemical modifications tell us the level of activity of these enhancers. So we can now use a second measurement in addition to measuring the real transcription. We can also use the chemical modifications at enhancers to predict the enhancer activity. And that's done using all these, genomics sequencing techniques. And something that I just want you to know is that you'll see these things like peaks. So these are each side as one enhancer, and these are sequencing reads. So at enhancers, for example, you have more signal, which results in more sequencing reads. So this is just to understand what those peaks mean. So, okay, so now you're almost expert in enhancer biology. Now you know what enhancers are, how they are regulated, what they do, and what's the structure of these sites. So what are the outstanding questions in the field? So one thing is people have been trying for so long to understand is where to find enhancers of DNA in the genome. You have 2 meters of DNA. Where can you find these enhancers? That's one. Can we predict enhancer activity based on DNA sequence? So I mentioned that we can measure the target gene expression to see how active the enhancer is, or we can measure the chemical modifications. But if I give you a DNA sequence and I tell you, okay, can you tell me how active this sequence is? Can you do that? That's the second question that the field has been trying to address. And then finally, once we have all this information, can we actually put nucleotides next to each other and make a synthetic enhancer de novo from scratch, right? That is the final goal of this field. And there's another point that I will not discuss today, which is, can we actually predict the target gene? So that's another unanswered question is, well, you have these regulatory elements, but which gene is regulated? And while there are techniques to address that, well, that's still an open question. So the first application of machine learning was to try to predict, based on DNA sequence, whether this sequence has enhancer activity or not. And the logic is you take different cell types and you profile, you measure the enhancers experimentally in those cells. Let's say cell 1 versus cell 2. And now you can use this machine learning, and now this is your expertise. I actually really don't know what means exactly, but this apparently is used a lot, the convolution neural network, for training the algorithm in a way that can recognize specific DNA sequences and can learn that these DNA sequences in cell type 1 are enhancers. And from there, so that's based on sort of pattern recognition. And the second data that fed into the model is the enhancer activity that I mentioned. So now you have two different things. One, the DNA sequence in particular cell type in which that enhancer is active. And the second parameter is the activity, the level of activity measured by the two techniques that I mentioned. And so based on this, the model now understands that this DNA sequence has enhancer activity. And now it can recognize which motifs should be present in this DNA sequence to have a stronger enhancer activity. What is the distance, for example, between these DNA motifs? And what's the combination? What's the orientation of these DNA motifs? So these are the parameters that are used by the model to then predict whether this sequence has, and to what extent, enhancer activity. Is that clear? So these are, again, the parameters that we are really interested in to understand, because you can have a combination of multiple motifs. You can have different order of these motifs, right? And you can have also different orientation, you can have different distance between these motifs, and all these will influence the activity of this particular DNA sequence. So that's actually what
this deep learning models are doing. So one example here, for example, they took enhancers in different cell types. In one cell type, and then they use all these chemical modification, all these measurements of enhancer activity, and they bind the entire genome into small sequences, and then they fed all these parameters, right? So now you have different beans, and each bean has multiple parameters. And then they train the algorithm in a way that recognizes, well, this bean with these particular parameters is likely to have enhancer activity. And so once they did this, they now took a different cell type and asked, can you use that training and predict the enhancers in this second cell type? And then they measured the sort of prediction experimentally. So that's all about predicting where enhancers are located. But in 2000, actually this year, two Nature papers were published and took this to the next level. And this is now the important thing. So once you train the model to recognize enhancers, you can then ask to generate a new enhancer de novo from DNA sequences. And that's what the sort of revolutionary step became. So there are different ways then to basically start from DNA sequence and generate a de novo enhancer. So they, in this, for example, paper, they use the Drosophila, the fruit fly, and brain cells. And then they use their deep learning model that they generated before, and then compared different cell types for their enhancer signature. So now they could say, for example, brain cells have enhancers with these features, whereas, for example, I don't know, skin cells have enhancers with these features. And so then basically they said, well, can we now start de novo from just a DNA sequence and train the model several times in a way that it will generate one enhancer based on all this information that if we test, we can show that has enhancer activity. So they started by this in silico evolution method, which basically they mutated nucleotide by nucleotide as a 500 base pair sequence. And it seems that 15 irritations is sufficient to generate one functional enhancer. And then they tested this with their previous model where they had basically a score for each active enhancer. And now they could actually sort of cross-check their synthetic enhancer with that score. And then they could score, for example, those sequences that have highest enhancer activity. And it's really nice because once they actually experimented, tested, 10 out of 13 of these synthetic enhancers functioned exactly as expected. So you can start now from nucleotides, basically. You can ask the program, generate a 500 base pair sequence that is functional in brain cells. And that is the super, super important thing. Now, the thing is that when you do this mutation, nucleotide by nucleotide mutations, you're basically generating motifs. So you're doing this mutation so many times that you can generate a small motif. So then they asked, well, what if you actually combine different motifs instead of starting from sequence? So that's what's called motif implantation. So here you can then combine different sequences which you know they are already motifs. So with this combination and inserting and deleting these motifs, now you can also generate a 500 base pair sequence which you think has an enhancer activity. And again, once they tested this, it was really nice because I don't think this is again 7 out this is sorry wrong. 7 out of 10 of those enhancers had enhancer activity. So this is really important because, now we can ask the computer to generate an enhancer for us that we can test. And the third approach, which is to me is the black box, I actually didn't understand it. They even didn't explain too much, is, well, they use AI now to generate these enhancers. I don't know how. The previous two approaches I could understand, but this, to me, it's a black box. But the same principle. So can you now make an enhancer de novo and test and show that it has enhanced activity? So this is super important because now for the first time, we can really predict why some enhancers are active in brain cells and why some enhancers are active in skin cells. And once you have this, it will open a lot of opportunities, right? So this is the final 2 slides. I'll give you just one application just to understand how important this concept is. So if you have a program that can make and synthesize an enhancer, well, you can do a lot of things with that. So for example, you can use that for gene therapy. So people now started to think, well, can we take, for example, tumor cells, normal cells, compare the enhancer pattern, use these deep learning models to recognize tumor-specific enhancers, right? And once you have that program, well, you can actually make an enhancer which is active only in tumor cells. So now if you use that enhancer to, for example, express a lethality gene, a cytotoxicity gene, right? That construct, that DNA, will only be active in tumor cells. So you can use this as a sort of tumor therapy, right? But that requires that you know the language of enhancer, you know how and which DNA sequence will only be active in tumor cells. So in the past, we had to test thousands of enhancers. First, we had to profile the tumor versus normal cells, and you still need to do that. And then we had to test thousands of these enhancer sequences to see which one is active in tumor cells. But now with all these models, actually you can predict. You can already make an enhancer which may not exist in real life. So the computer may come with a sequence which does not exist. It's a combination of all these sequences from different enhancers. But that is an enhancer that is super active in specifically your tumor cells that you can use it then for gene therapy. So that's what we are doing in my lab. So we are trying now to understand this in chemotherapy-resistant tumor cells. So one of the major problems in cancer therapy is that tumor cells become resistant after some time. So we are now trying to address what are the enhancers that are specifically active in chemotherapy resistant cells. And once we find those, we can actually use the same gene therapy approach that I mentioned. So we did this, we profiled these enhancers, we found 2,000 enhancers that are active in drug-resistant tumor cells. And now we are measuring their enhancer activity using the technique that I mentioned. Once we have that, then we can basically train a model in which can recognize the pattern of enhancers that are active only in drug-resistant cells. And that is the rate limiting factor. Once we have that, from there it's much easier. So we can basically generate these constructs, use them for gene therapy, to target cells that are resistant to drugs, and to basically prevent or overcome chemotherapy resistance in cancer. Okay, so I will stop here. Again, this was mainly to give you a sort of high-level overview of what other applications can be used using AI. And it was mainly on, again, the genomics, enhancer biology, and the language of the enhancer code.